Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my last lecture on biomolecules. So today we're going to say a big goodbye to biomolecules. So we're going to finish off the smallest bit that we have, and then we're going to discuss some important questions. So I have almost 20 questions which we'll discuss, and then we will end this chapter. So Monday you'll have the Menti quiz from this chapter. All right? Yes. So shall we go forward? So can we finish off this chapter, the little portion that we have? Yes. So in the last lecture, so that is on Wednesday, so I've been discussing about enzymes. So how an enzyme works to catalyze a particular reaction. So what are the factors that affect an enzyme, and so on and so forth. So today we're going to talk about the nomenclature of enzymes and what is this concept of cofactor. Alright, so these are the little things that is left, so we'll finish it off quickly. So before that, I will definitely give you a recap of what I've been doing in the previous class, right? So we were talking about what are enzymes. So how powerful are the enzymes, right? So we know an enzyme binds to the a substrate binds to the enzyme in its active site. So an enzyme can have more than one active site, right? So this is my enzyme, this is my substrate, and this is my active site. Okay, right. So when an enzyme substrate binds to the active site of an enzyme, so there undergoes a conformational change. So you have the enzyme substrate complex, which very quickly changes to form your enzyme as a product complex, and it gives out your enzyme and your product separately. So this enzyme substrate complex is transient and unstable. Okay, so what are the factors that affect the enzyme enzymes in a reaction? First is your temperature. Next is your pH. The third one is your substrate concentration. Right? So every enzyme has an optimum pH. So every enzyme works well at a particular optimum pH, and similarly every enzyme works well at a particular optimum temperature. So if for example, if the optimum temperature is 37 degree and if I bring the temperature less than 37 degree, so at lower temperatures, so the enzymes get inactivated temporarily. Okay, so at high temperatures they are denatured. Fine, clear? So now talking about the last one, about substrate concentration. So we read this graph. So substrate concentration, this is the velocity of the reaction. So as the substrate concentration increases, the velocity of the reaction increases. So when there is maximum substrate concentration, the velocity is maximum, which we call it as Vmax. But now what is the substrate concentration that is needed to attain half Vmax? So half Vmax is half maximal velocity. So what is that concentration? So that is your Km value, that is Michaelis-Menten's constant. So this is where we spoke about the concentration of substrate. So the last concept was about inhibitors. So inhibitors inhibit the process of enzyme substrate process, or it inhibits the work of enzymes in the entire reaction. So inhibitors can be of two types. One is competitive inhibitors. The other one is your allosteric or feedback inhibitors. So in case of competitive inhibitors, the substrate and the inhibitor, they compete for the active site. They are identical in structure and they compete for the active site. So here the example can be Succinate is our original substrate. So malonate is our inhibitor. So the enzyme here is succinate dehydrogenase. So the next type of allosteric or feedback inhibition is when the product inhibits the enzyme. For example, when I say A gives me B in the presence of enzyme C, so B itself will inhibit the work of C. So when more amount of product molecules are formed, so that itself will inhibit the formation of the substrate. So example is your glucose to glucose 6-phosphate. So this is catalyzed by the enzyme hexokinase. So your glucose 6-phosphate, when it is more in amount, so it can inhibit your hexokinase, thus halting the process. Alright. Okay, so this is what we discussed in the previous lecture. So now moving forward to the nomenclature of enzymes. Right, so how do you name the enzymes or how do you classify them? So you have a lot of ways, right? So what are the ways? So there are 6 ways by which you can classify enzymes. The first way is oxidoreductases, alright? So oxidoreductases are also known as dehydrogenases. So then you have transferases, hydrolases, lyases, then you have isomerases and ligases. So these are the 6 ways by which we can classify enzymes. So let's go one by one. So first let's talk about oxidoreductases. So they catalyze oxidation reduction reactions. So they catalyze oxidation reduction reactions, that is they help in the catalysis of the transfer of electrons, right? Yes ma'am, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. So they catalyze the transfer of electrons, right? So there are 3 types of oxidoreductases. First is oxidases. Then you have dehydrogenases, and number 3 you have reductases. So example of oxidases is your cytochrome oxidase, of dehydrogenases, which we just read now. So that is succinate dehydrogenases. So reductases, you have your nitrate reductase. So these belong to this class of enzymes, that is oxidoreductases. Alright. So the next one is transferases. This catalyzes the transfer of the other functional groups. Okay. The second one is your transferases. Say for example, I have one substrate and it has our group, side group, right? Then it has another substrate. So now it will be like this. So the G has been transferred here, right? So now the group should not be in its free state. So then the transfer will not happen. So it cannot be like this. No, the functional group that you are transferring should be in the conjugated form. It should not be in the free form. So this is your transferases enzyme. Alright, clear? Fine. So the next one is hydrolases. It breaks the bonds. Okay, so that is your hydrolases. So it catalyzes the splitting of larger molecules to form smaller ones. So I will write here hydrolases. Okay, so these hydrolases, they catalyze the splitting of larger molecules to smaller ones. So this is what hydrolysis does, okay. So this is basically hydrolysis of water. So by the elimination of water, so they have this process, right. So let's say starch is converted into maltose. So you have the amylase, right. So this breaks down the bond that is present here. So this is one type of your nomenclature. The next one is lyases. This is number 4, that is lyases. So lyases, it breaks specific covalent bonds. So this catalyzes the breakage of specific covalent bonds. So that is broken down here. So for example, if I have, so it will form like this. So this is what your lyases does. So number 5, so you have your isomerases. So this is based on the specific position or rearrangement, right. So you have glucose 6-phosphate to fructose 6-phosphate. So here you have your phosphohexose isomerase. So they have the same number of carbon, glucose and fructose, but the arrangement is different. So now talking about the last one, that is ligases. This joins 2 or more bonds. So that is your ligases. Alright. So far clear? So what does ligases do? So joining of 2 substrate molecules. So this basically joins. So this is about the nomenclature of your enzymes. So now moving forward to what are cofactors. So enzymes are mostly proteins with the exception of ribozymes, right? So enzymes have a protein part as well as a non-protein part to make it a complete portion, right? So every coin has 2 sides, the head and tail, right? So similarly, enzymes have the protein part as well as the non-protein part, which makes it a complete enzyme. Alright, so here we go. So the protein part of the enzyme is called as apoenzyme, the non-protein part is called the cofactor, and the whole enzyme is the holoenzyme. So holoenzyme is equal to apoenzyme plus cofactor. So apoenzyme is the protein part and cofactor is the non-protein part. Fine? Okay. So this cofactor can be of 3 types. So this can be of 3 types. So the first type is your prosthetic group. The second one is your coenzymes. And your third one is your metal ions. So your prosthetic group, it binds very strongly, or it is tightly bound to the apoenzyme. So apoenzyme is the protein part, right? So it is tightly bound to the apoenzyme. Alright. So the prosthetic group, for example, heme is the prosthetic group, right? So your hydrogen peroxide is broken down into water plus oxygen, right? So you use peroxidases or catalases. So these enzymes have your heme as the prosthetic group. All right? Yes? Fine. So now talking about the coenzymes. So these are transiently bound to the apoenzyme. So unlike the prosthetic group, these are not tightly bound. Mostly the center portion of your coenzymes are soluble vitamins like B and C. So you have your NAD, FAD, FMN, right? So these are your coenzymes. The next one is metal ions. So metal ions form coordination bonds on one side with the substrate and the other side with the apoenzyme portion of the enzyme. So this is what your metal ions are. For example, Zn2 plus is the metal ion for your carboxypeptidase. So it forms coordination bonds on one side with the substrate and the other side with the apoenzyme. So with this we come to the end of the entire chapter of biomolecules. So let's see how much you guys have understood. So I have 20 questions here. So now we'll discuss those 20 questions so that you can let me know if you have any doubts. So if you're understood, if you're able to answer the questions, well and good. If not, so please pay attention. This is how the questions can come. So the turnover number of enzyme depends upon the size of the enzyme molecule, the number of active sites, the concentration of the substrate molecule, molecular weight of an enzyme. So here I am not talking about the turnover number of a biomolecule, but instead I am talking about the turnover number of an enzyme. So an enzyme is quickly converted into enzyme substrate complex and then into enzyme product complex. So it is changing from one form to the other form. So the turnover number of the enzyme is based on which of the following: size of the enzyme molecule, number of the active sites, concentration of substrate molecule, molecular weight of an enzyme. So this is based on the number of active sites that is present in an enzyme. So again I am repeating, an enzyme can have more than one active site. Alright, so here we go to question 2. The Km value of the enzyme is the value of the substrate concentration at which the reaction reaches half maximal velocity. Right, so I drew the graph just now while giving you the recap. So substrate concentration, velocity. When there is maximum substrate concentration, that is Vmax. So the half maximal velocity that is needed. So the substrate concentration at this point is your Km, right? So here the answer is C. So feedback inhibition of an enzyme is affected by the end product. So when A gives me B in the presence of the enzyme C, B itself will inhibit C. Right? So the inhibition is happening by the end product. So glucose to glucose 6-phosphate catalyzed by hexokinase. So this hexokinase is inhibited by glucose 6-phosphate itself. Right? So question 4, which of the following statement is correct with reference to enzyme? So I just taught you. So holoenzyme is equal to apoenzyme plus coenzyme. So coenzyme is equal to apoenzyme plus homoenzyme. So which one is right? So here you have to tell me which is the protein part and which is the non-protein part. Yes, quick. So your holoenzyme is apoenzyme plus your coenzyme. This is your right one. So coenzyme is a type of cofactor, right? So question 5, the relation between amino acid and protein is similar to the one found in. So polymers of amino acids make up protein. So I have many amino acids. So this many amino acid makes up my proteins. Right? So similarly, one nucleotide, the second nucleotide, the third nucleotide. So polymers of nucleotide makes my nucleic acids. So proteins and amino acids, it's similar to nucleic acid and nucleotides. Right? So question 6. So antiparallel strands of a DNA molecule means that, so if I draw the DNA like this, so I have 5 prime, I have 3 prime. I have 3 prime, I have 5 prime. So we have phosphate in the 5 prime group and in the 3 prime group we have the OH, right? So which means the phosphates are, the phosphate groups at the start of the 2 DNA strands are in the opposite position because we have an antiparallel polarity of your DNA molecule. So all enzymes are proteins, right? But this statement is now modified because of one exception. That one exception is ribozyme because this is a nucleic acid. Fine. So we go to question number 8. Select the type of enzyme that is involved in the following reaction. Yes, sir. A very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So I have the reaction here. So I have one substrate that is conjugated with with G and another substrate S dash. So now if you can see, the group is getting transferred here. So this belongs to a group of enzyme family that is called as transferases, which I just taught you, right? So here it is option B. Alright, so we go to the next one, question 9. Transition state structure of the enzyme formed during an enzymatic reaction is, so the enzyme substrate complex is the transition state complex, right? So this transition state complex is transient and unstable. So transient means it is formed for a short time and unstable means it is about to degenerate. Okay, so we go to question 10. For a protein to have a quaternary structure, that is the fourth level of the protein structure, so it should have at least it should consist of 2 or more polypeptide subunits, right? So it should have many subunits of the tertiary structure to become the quaternary one. So am I right? Yeah. So we go to question 11. The quaternary structure of human hemoglobin is best described as a tetramer with 2 different subunits. So tetramer means 4. But we have 2 different subunits, 2 alpha and 2 beta subunits. So this is how best it is described. Alright. So question 12. At a temperature near the freezing point, the enzymes are inactivated. So at lower temperature, the enzymes are temporarily inactivated, and at higher temperatures, they are denatured. So hexokinase glucose to glucose 6-phosphate belongs to the category of which of the following? So here an additional phosphate group. See, so phosphate, here it is triphosphate, right? And here it becomes diphosphate. So phosphate group is getting transferred from here to here. So they belong to a family of transferases because they are transferring the phosphate group. Alright, so question 14, what will happen when the cofactor is removed from the enzyme? So the cofactor is the non-protein part. So when the non-protein part is removed from the enzyme, so the catalytic activity of the enzyme is lost. So your enzyme will not be as efficient as it should be. So coming to question 14, the primary structure of a protein polypeptide is stabilized, or the secondary structure of the polypeptide is maintained by hydrogen bonds, right? So that is very important. So that is what it drives. Right, so here peptide bonds is not given, so please don't get confused. Ma'am, it's polypeptide, so it should be peptide, not necessarily. So question 16, the catalytic efficiency of 2 different enzymes can be compared by the Km value. So because Km value gives us half maximal velocity. Right, so when I say 2 is the Km value, that is giving me half maximal velocity, and for another enzyme it is 4. So it is enough to compare which enzyme is efficient, right? Yes. So question 17, enzyme that breaks nucleic acid into nucleotides belongs to which of the following? So that is so here we have to break nucleic acids into nucleotides. So this came in your digestion as well. So here it would be hydrolysis. It is breaking the bonds. So your bigger molecules are broken down into smaller ones, right? So hydrolysis catalyzes the splitting of larger molecules to smaller ones. Okay. So the last 3 questions. When triglycerides are solid at ordinary room temperature, they are fats. Right, so they are solid at ordinary room temperature. So you have your animal fats like butter sometimes, right? So all these are present. So question 19, when glucose is degraded to lactic acid in the skeletal muscles, energy is liberated. So which of the following conclusion is correctly associated? So energy is liberated in a catabolic reaction. In an anabolic reaction, energy is utilized. So here when the statement says energy is released, so it is undoubtedly your catabolic reaction. Alright. So the last question, the stored form of sugar in animal is your homopolysaccharide, that is your starch, right? So this is the right answer. So with this, we have finished the chapter of biomolecules and we have also completed 20 questions discussion. So on Monday, we have Menti quiz from the same chapter, that is biomolecules. So which chapter to start next? So please tell me in the comment section so that I will start with the next chapter very soon or next Wednesday. So I hope you have understood biomolecules really well. So I have divided into 7 lectures and I have taught you and we have even discussed questions. So please let me know if you guys have any doubt and if you genuinely like my way of teaching. So please do like, share and subscribe. So I'll meet you on Monday with a very interesting Menti quiz. So until then, have a beautiful weekend ahead. I'll meet you. So bye, take care and stay tuned. Bye.